Hi, everyone. Thanks for attending our webinar today. My name is Helena Mistry, and I'm the Director of Business Development at IMICA. Today's webinar with Guido Guberman, our speaker, will be a 30-minute presentation by Guido, followed by 20 minutes of Q&A. So at the end of his presentation, if you would like to ask a question, you can click on the raise hand button and I will unmute you, or you can type it into the question box and I will read it out to Guido. So the total time of our webinar today is 50 minutes. At the end of our webinar, we'll send out a brief questionnaire or survey. So if you could fill that out, it would be helpful for the planning of future webinars. And we'll be recording today's webinar. So the title of our webinar today is Concussion Biomarkers, Novel Imaging and Advanced Statistics. A little bit about our speaker today. Guido is currently an MD PhD student at McGill University in Montreal, Canada. His work consists in combining advanced statistics with novel diffusion MRI approaches in order to improve diagnostic and prognostic criteria of pediatric concussions. Guido collaborates closely with Maxime Descoteaux, IMICA's CSO and professor at the University of Sherbrooke in Quebec, Canada. In the future, Guido aims to specialize in pediatric neurology and become a clinician scientist. Okay, Guido, I'll let you take it from here.
Good. Thank you, Helena, for that introduction. So I'm just going to start my presentation. Can you give me a confirmation that you can see this, maybe?
Yes.
Okay,
great.
My mouse as well?
Yes.
Okay, perfect. All right. Okay, so I'll start now then. So first off, just want to thank IMICA for inviting me to speak. And let me know about any point during the presentation you can't hear me very well or anything happens, all right? So my work is on pediatric concussions. If anybody here is familiar with the field of concussion research, then perhaps the word that comes to mind is frustration. Frustration because despite millions of dollars invested in basic science and clinical trials, there has been little to no progress in terms of the pharmacotherapy of concussions and really clinical management as a whole. I think this wonderful review by Erin Kenzie and colleagues really summarizes it well, especially the part that I highlighted here. It paints the picture of a field that is in a severe state of stagnation. There are many reasons why concussions are challenging, far too many for me to cover in the scope of this presentation, but I'm going to be focusing on one which I believe is perhaps the most important challenge, and one which has only now started getting the attention that it deserves. So I'm talking about concussion heterogeneity. True to the definition of the word, concussion heterogeneity refers to many things, in that concussions affect many people who are very different in very different ways. So what I mean by that is that there are factors to keep in mind, like genetic predispositions, sociodemographic factors that play a role in pretty much every level of concussion. There are pre-injury comorbidities to keep in mind. In terms of, well, at the level of the brain, concussions elicit several different neuropathological cascades, not all of which occur in sequence. Some might occur in parallel, some might occur in some people and not others. And even for the people who have the same neuropathologies, these don't occur necessarily in the same time frame. In addition, pretty much any area of the brain could conceivably be affected. And if we look at concussions just clinically, you notice that, for instance, if you take the most well-known concussion symptom scales, you see that they have upwards of 20 symptoms, which not a whole lot of other diseases have that many different, seemingly unrelated symptoms. So symptoms can be very variable. Their severity can be very variable as well, and so is their duration. So it's no exaggeration that concussions are extremely heterogeneous, and as a result of that, they are extremely challenging. Now, what about heterogeneity is so problematic for concussion research? Well, it comes from the fact that the vast majority of concussion studies, basic science, and clinical trials they all use the same study design, which is, you can call it a group comparison design. So it goes something like this. Imagine that you are interested in doing a brain imaging study to compare concussed against healthy controls for the purpose of developing some kind of diagnostic biomarker. So you take your concussed group, you scan them, you take your healthy controls, and then you sort of calculate some average concussed brain, some average healthy control brain. You compare the two, and whatever you find to be different between the groups, oh, there's our diagnostic biomarker. Now, we could have replaced these labels by anything that you could think of. You could have said symptomatic concussed versus asymptomatic concussed. You could have said concussed with headaches versus concussed with non-headaches. Really, and you could have said anything. And clinical trials sort of work this way as well. Now, the problem with this approach is that it relies on two very important assumptions. The first is that whatever is shared across the group is way more important than whatever is less shared or more idiosyncratic. Or another way of saying is, it's okay to just average out heterogeneity. This is what the assumption relies on. The second assumption is that there exists a consistent, mostly one-to-one mapping between symptoms and brain structure or function. So what do I mean by this? Say that you have three people who have sustained a concussion and at some point report suffering from sleep problems. You might, pretty reasonably so, conclude that these three people have damaged areas of the brain controlling or related to sleep. This assumption is not, as I said, it's very reasonable. In fact, you wouldn't be alone in making this assumption. Most of the field makes this assumption. But the reality is that perhaps that's not entirely true. It's been theorized that brain structure and function and symptoms, they can interact in very complex ways. So here's what I mean by this. Going back to our example, one of these people might have indeed damaged the area of the brain related to sleep. But the second person may have damaged the areas of the brain related to mood control. This person might develop a depression, and a common symptom of a depression is sleep problems. To take another example, this person may have damaged the areas of the brain related to attention control. That might lead them to have a very hard time focusing on their job. They might get fired as a result. And the stress that comes from not having a job could have led them to experience sleep problems. So you have three people who have pretty much the same problem, well, the same condition, by that I mean the concussion. They have a common symptom, the sleep problems, but these three people are clearly suffering from very different problems when you look at them in detail. So the objective of the study that I'm going to present today was to parse concussion heterogeneity or account for concussion heterogeneity, but in a way that can account for these complex relationships between different symptoms and different brain structural problems. And we like to call these things multi-track, multi-symptom relationships. That's the term that we gave these things. So to do this, we took data from the Adolescent Brain Cognitive Development Study. This is a multi-site study out of the US. It has data for close to 12,000 children. And a sizable proportion of them have had a history of a previous concussion. Now keep in mind that these are cases of, most of these cases are cases of chronic concussions. They're well over a year since their injury. After dealing with issues with the data, we were left with a final sample of 306. So these children had lots of interesting data. What we used was multi-shell diffusion MRI data, and here are the acquisition parameters for anybody who's interested. They also had T1 weighted images, and they had behavioral measures from several different questionnaires that we used to probe different potential symptoms of concussions. So we processed all the data using Track2Flow, which is a processing pipeline developed by Max's lab. And here I'm just summarizing my post-processing pipeline. So what we did after we processed the data. So we took our whole brain tractograms that we got from TractorFlow, and we combined them with a brain parcellation called DKT parcellation. And that gave us a big matrix of which areas of the brain are connected to which for all of our participants. We then, as an aside, I'm just going to tell you that you might notice some technical details that I'm skipping for the sake of simplicity. If you're interested in them, please let me know. We can talk afterwards. So after that, we selected some white matter connections, and on those connections, we took several measurements. Now, we took some measurements that perhaps you might be familiar with, things like fractional anisotropy, or FA, axial diffusivity, mean and radial diffusivity. So these are measures that are based on a pretty conventional way of modeling diffusion MRI data called the tensor model. Now, we also used another approach, a more novel approach called constraint spherical deconvolution, which is a modeling technique that is capable of, leaving the technical details aside, it is capable of addressing some of the limitations that the tensor model used to have. So as a result, the measurements that come from this model, apparent fiber density along pixels and the number of fiber orientations, these two measures are less biased by the same problems that the previous measures were. And as a result, they are much more informative or much more specific. So I'll take a second to walk you through what some of these measures mean. As I told you before, concussions elicit different neuropathologies at the level of the brain's white matter. And here I'm only showing you white matter, but if I showed you gray matter as well, it would be even more complicated. But what you have to notice here is that these classical measures like fractional anisotropy, they are very sensitive. They can detect differences in microstructure due to several different things, some pathological processes, some non-pathological processes. But the problem is that their specificity is very poor as a result. They're affected by everything and anything, really. Whereas, as you can see, some of the more novel measures like apparent fiber density along pixels, they are much more specific. And as a result, they provide a lot more information. Now, that being said, none of these measures by themselves are perfect. They all capture different aspects of the underlying microstructure. And I like this metaphor of it's as if several blindfolded people were feeling different parts of the elephant and guessing what the elephant was. So these microstructural measurements, that's what they do. They capture different aspects of the underlying true microstructure. Some things might be overlapping between these measurements. Some things might be unique. Now, there was an approach developed recently by a previous student from Max's lab that uses principal components analysis, or PCA, to mix and match these different measurements into fewer but much more informative measurements that ultimately try to capture all those shared things about brain microstructure that these individual measurements are finding. So that's exactly what we did. We performed a PCA on all of our connections and across all of our measurements. And jumping ahead to some of our results, I want to show you that we found two measurements that were highly informative, and that they accounted for about 97% of the variance across all measurements. So these are highly informative measurements. Now, what do they represent? So the first one, based on the pattern about how it represents different symptoms, we can conceive of it as a measure of microstructural complexity, in that the lower the score for this measurement, the more simple or the less complex the microstructure is, and vice versa. The higher the score, the higher the complexity. The second measure, as you might have seen, is much more specific towards axonal density. So we can call this one a measurement of axonal density. Now, we took these two measurements and we fed them into what is really the workhorse of this study, which is this algorithm here called partial least squares correlation, or PLSC. So PLSC is a cousin of PCA. It does pretty similar things. It mixes and matches variables into much more informative variables. But the crucial difference is that it does it on two variable sets at once. So it takes the X variable set, mixes and matches the X variables into one highly informative, one or fewer than or a little more than one, much more informative measures. It does the same thing on another side. It takes Y variables, mixes and matches them, puts them into an informative, highly informative measure. But the very important difference as well is that it is creating these informative measures in such a way that it maximizes the relationship, or in statistical terms, the covariance between these two measurements. So that's exactly what we did here. We took our connectivity measures, we took our symptom measures, we fed them into this algorithm, and what came out were these pairs of, on one side, multi-track connectivity features, on the other side, multi-symptom features. Together, these formed what we call multi-track, multi-symptom pairs, or multi-track, multi-symptom relationships. And that's what we were interested in. So I hope that was clear. Now we're going to go on to our results. One of the main things we wanted to do in this study was to compare our novel technique against more traditional ways of analyzing this data. So the first thing we did is that we computed very simple classical correlations between the connectivity measures across all the white matter connections that we studied and all the symptoms that we had. So we had a massive correlation matrix of every single possible combinations of single track and single symptoms. And this is what you're seeing here in blue, the distribution of correlation strengths or coefficients. On the other side, we took our multi-track connectivity features and we took the correlation with their corresponding multi-symptom features. And that's what you're seeing here in pink. So what I wanted to notice here is that all multi-track, multi-symptom relationships were much stronger than all single-track, single-symptom relationships. Or another way of saying it, they were much more informative. They captured much more information. Now, this is interesting, but not terribly surprising. These multivariate statistical analyses, this is what they do. They find these hidden, very strong patterns. So we wanted to go a little deeper and explore even more this question of this comparison between traditional versus our novel approach. So we had, for the measurements, behavioral measurements that we had, came from this well-known behavioral checklist called the Child Behavior Checklist. So these were the measurements of depression, aggression, anxiety, and attention problems. So there's a well-known, pretty standard cutoff that if you have a score above 70 in any of these scales, you have a clinical range of whatever problem you have. So what we did is that we took everybody who had a score above 70 in any of these 4 scales, and these people were the individuals who had clinical levels psychopathology. So that allowed us to compare a clinical level psychopathology group against a non-clinical level psychopathology group. So what we did is that we took all the connections, all the white matter connections that we were studying, and we compared their level of microstructural complexity for every single one of these connections, using pretty standard univariate analyses. So that allowed us to identify these connections that you see here in red in this chord plot that are significantly different between these two groups. And the classical interpretation, the way that these connections would be interpreted would be psychopathology related connections, right? Because they're different between this psychopathology group and this non-psychopathology group. What we wanted to do was to look at how many of these connections were also significant in our multivariate features that we built. So this is what you're seeing in this plot. It's the percent overlap between our multivariate features that we build and the univariate analyses that we ran between the psychopathology and non-psychopathology group. And what you can see is that the percent overlap is much higher for the first multi-tract, multi-symptom feature than all the other multi-tract, multi-symptom features. Now, this might seem a bit abstract to you, so I'm gonna walk you through the interpretation because this is a very important result. So let's take our first multi-tract, multi-symptom pair. We call it the first because it's the one that accounts for the highest covariance. This is what you're seeing in these bar graphs here. When I say that it accounts for the highest amount of covariance, in other words, what I'm saying is that it's the one that is the most consistent across the group. This is what is most shared across people across the concussed people that we studied. When you look at the symptoms that it represents, you see that it represents most symptoms more or less equivalently and all in the same direction. So the pattern of the way that it represents symptoms led us to take some liberties and call this pair a general problems pair. So it reflects whether you have any issues after sustaining a concussion. What I want you to notice here is that 23% of the psychopathology related connections were also identified as general problems connections. Now, this is not terribly surprising. When you have general problems, or rather when you have psychopathology, it makes sense that you have, that's part of general problems, right? But let's take another example. This is our multi-tract, multi-symptom pair number 4. As you can see here, it accounts for less covariance, which another way of saying it is that it is more idiosyncratic. It is less shared and consistent across the group. Based on the way that it represents symptoms, based on the sign and the magnitude, we can say that this pair represents psychopathology a little bit more specifically than our previous pair. And again, taking some liberties, we can, and I know this is an oversimplification, but we can call the connections that came in this pair psychopathology-related connections. What I want you to notice here is that only 4% of the connections that we identified as psychopathology-related connections based on our traditional group comparison, only 4% of them were identified as psychopathology-related connections when we did this multivariate analysis. So that raises the question, what are these connections that we identified or that are identified based on traditional approaches? Are they really psychopathology-related connections, or are they perhaps reflective more of general problems? Now, to make matters more complicated, you may have noticed that these chord plots contain these black lines within them. These lines actually represent connections that pass through the corpus callosum. And what I want you to notice here is that when we did this traditional group comparison, we found several connections that pass through the corpus callosum. And again, remember, these would be interpreted as psychopathology-related connections. Looking at our multi-tract, multi-symptom feature 1, remember, this is the general pair, the general problems pair. You see that a lot of connections also implicate the corpus callosum. However, when we look at our psychopathology-related pair, much fewer connections implicate the corpus callosum. So if we put all of these results together, this is the interpretation that we have. When people sustain concussions, they have these complex multi-tract, multi-symptom relationships. These might be sort of idiosyncratic, and they might not necessarily be shared across people. So this is the kind of stuff that we observed with our multi-tract, with our psychopathology-related pairs. When you pool across a group, you get rid of all the stuff that is idiosyncratic, and you just keep the stuff that is consistent across the group. The kind of stuff that you see in our general problems pair, the most consistent things. Now, for reasons that are not entirely clear yet, a lot of these things that are most shared across the group seem to implicate the corpus callosum. And the issue here is that you are going to be, whoever does the study where you pool across people, you're going to be inferring a relationship between whatever connectivity feature you found to be significantly different between groups and whatever contrast you had between groups. Let's say that it was concussed, non-concussed. Let's say that it is people with sleep problems versus people without sleep problems. Now, the issue is that you've just inferred a relationship that really was just the result of your group comparison design. And that might be the reason why, if you're at all familiar with the field of concussion, you will have seen this. The corpus callosum is implicated in everything and anything. It seems to come up as a significant difference between concussed and non-concussed, symptomatic concussed versus non-symptomatic, cognitive problems versus non-cognitive problems. It's everywhere. And it's a chance that this might just be the result of this traditional group comparison approach. Now, at this point, you might be wondering, so what? It seems like this is a pretty esoteric set of results. If you are a concussion researcher, this is why you should care. The field of concussion research, as you might be aware, is moving towards these very large-scale studies, coming from international multi-site collaborations, these amazing studies in which their sole purpose is to acquire lots of data and then pool it. They want to try to pool all this data together because most people in the field of concussion, they see the problem of concussion heterogeneity as one of statistical power. So what they want to do is they think that if they just pool enough people together, they can get rid of all the heterogeneity and finally find that elusive concussion biomarker. In other words, they want to do more of the same, just on a larger scale. In this respect, these results are fundamentally different. Because what we're proposing essentially is a paradigm shift. We're saying that the issue with concussion heterogeneity comes from the fact that really it's in the heterogeneity that the interesting things are. As they say, the devil is in the details, right? And instead of trying to regress out or average out concussion heterogeneity, we should embrace it. We should leverage it, because perhaps that is where we can move forward. Now, if you're a clinician or you are part of the clinical world, you might still be wondering, so what? I'm a clinician training myself. I understand that you might be wondering, how does this impact my work, my clinical work? How does it impact my clinical practice? So let me leave you with one final set of results. In the same way that you can look at the expression of a gene, you can look at the expression of these multi-tract, multi-symptom features that we built. And that's what we're showing you here in these scatter plots. In black, we're identifying those individuals who had clinical level psychopathology. Now, I want you to notice that, I want you to think about the fact that these people with clinical level psychopathology, in most studies, in most traditional studies, and certainly in clinical trials, they would be grouped together, they would be lumped together. But as you can see, when you look at the expression of these multi-tract, multi-symptom pairs, these people are quite different. They're quite differentiable. You can see it for our general problems pair. You can see it for our psychopathology related pair. And the reason why they're so differentiable is because what this approach does is that it can find patterns of combinations of different symptoms and combinations of different brain abnormalities. So in the future, and at this point we get into speculation, but in the future, once we validate these further, once we study them further, what we want to do is we want to see if perhaps rather than stratifying by very simple things like sleep problems, yes, no, headache, yes, no, concussed, not concussed, we want to see what happens if we stratify based on these multi-dimensional features that we built here and see if this is what finally moves the dial forward in terms of concussion pharmacotherapy and clinical management. So I'd like to thank the people who collaborated with me on this project. I'd like to thank people who contributed advice or code that was very important for this project. I'd like to thank funding agencies, and of course, I'd like to thank once again IMECA for inviting me to give this lecture. If you are at all interested in finding out more, there are several other results that didn't have the chance to present today. This paper is up on medRxiv as a preprint, and we will be submitting it soon. So you're welcome to take a look and provide any feedback. So thank you very much for your attention, and I look forward to your questions.
Thank you very much for that, Guido. That was a great presentation. I would like to open this up to questions, and I have my first question from Aina Fra Pascal. Aina, go ahead.
Can you hear me?
Yes.
Yeah. Thank you very much for your talk. It was great and very, very interesting work. I was wondering, when you were talking about the corpus callosum, that you find consistent differences there. Because we know that the corpus callosum is very sensitive to, for example, motion, and to like differences in the methods, like how you mask your data and all this stuff. And I was wondering if you accounted for that, or like at least for motion, or if you checked the data.
Yeah, so the way that we process the data, this is part of that pipeline that I mentioned, track of flow. At some point, there's a step in which the data is motion corrected. But in addition, one thing that I also skipped is that the ABCD study themselves, they provide, even before releasing their data, they perform their own quality checks. They release the score from their quality check. So all this data was quality checked by the ABCD study group, and also it was checked by us after processing. So, you know, after processing, we actually did a visual inspection of 306 images to account for stuff like this, like motion and all that.
Okay, thank you. And I was wondering also if you have considered using different atlases for the,
so that's a fair question, and the answer, I mean, we might like to. As I mentioned at some point at the end, we want to, when I said that we want to validate these features further, that's exactly what we mean by that. We want to go at every single node of our study and look at, you know, what are the other options? What happens when we try other options? That's what we mean by validating. So
when I was listening to your presentation, there are so many things that you can do with all this analysis that you've done.
Yeah,
but I want to congratulate you, because it's a great work, and I'm looking forward to reading your paper.
Thank you.
Thank you. Guido, I have a question. I noticed that you focused on diffusion MRI. So could you comment on the importance of white matter with concussions versus gray matter?
Okay, that's a big question. Thank you for that. So depends on who you ask, because there's people who are very invested in studying each one of these different areas. But it's pretty well known within the field of concussion research that white matter is very, very sensitive to concussions and to traumatic brain injuries as a whole across the spectrum. And it comes from the fact that every time that you have a brain injury, your brain essentially just like wobbles inside your skull. And there's a lot of shearing forces that are transmitted across the brain, and white matter is very sensitive to that. It's been, by some pretty big names in the field, concussions have even been called a disease of disconnection, or a disease of white matter connection. I think that's perhaps taking it a bit too extreme, but it is extremely important to study white matter in concussions for certain.
Okay, great. So thank you for that. Our next question comes from Sammy Obeid. Sammy, if you'd like to go ahead and ask your question.
Yeah, thank you. First of all, thank you, Guido. Great talk, extremely interesting. And I believe you can apply your methodology to many different diseases, not only concussions, but very well done. So I have a few questions, if you don't mind. First question, how were the patients included in your study? In the sense that concussions, as you mentioned, is very heterogeneous. What criteria were used to include patients and to consider them concussed?
Mm-hmm. Okay, so the answer to that question is that the ABCD study has pretty serendipitously acquired not only questionnaires about a history of concussion, but the amount of time that they spent, that they lost consciousness for, the amount of time that they had amnesia for, and really it's based on these criteria, which perhaps you might have heard of the WHO criteria of concussions. Mm-hmm. That's what we use. So loss of consciousness for less than 30 minutes, post-traumatic amnesia for less than 24 hours. And additionally, none of these people had any CT findings or anything that might move them into the category of complicated or complex concussions.
Okay, so you use the WHO criteria.
That's right.
So they used to include patients as concussed. Okay.
That's right.
Second question I had is, you mentioned obviously there were differences involving the corpus callosum. As you know, the corpus callosum is extremely vast in the brain. Did you find any pattern or more subregions of the corpus callosum that were involved in your results, or was very let me put it this way, was it more the parietal corpus callosum, the frontal corpus callosum, the one that's located to the splenium, the genu, and so on?
So one of the consequences of the parcellation that we use is that we don't have these clear-cut and I would say well-recognized divisions of the corpus callosum. That's why I was very careful in my words. There were tracts that pass through the corpus callosum, right? So these are tracts that if I put all these tracts together, they would form the corpus callosum, but they're not like anterior, like genu, midsection, and all that. Are there any patterns? Keeping that in mind, I could go look at what corpus callosum tracts I had, and I would like to see any patterns. So that's a great idea actually, to go see if there are any patterns. I didn't check for any patterns, but as I said, any patterns that we find are not going to be that clear cut, because they're big sections of the corpus callosum.
Because maybe you could maybe relate a certain portion of the corpus callosum with certain symptoms.
That's a fair point. One thing that I would caution though about that is that I want to say this carefully. It's kind of dangerous to use these multivariate analyses and then go look at individual symptoms. That's something you have to be careful, because the relationship between them, the connections and the symptoms is not that clear cut. It's not like, yes, we have damage here, therefore we have that explains this symptom. It takes a little bit more work than that. It's a little bit more complicated than that. But certainly it would be worthwhile to look at patterns within them for sure.
And finally, how do you see your methodology and your whole the whole project you did as? Would it be useful in orienting treatment, in more of for diagnostic purposes, or more treatment purposes, and so on?
So it depends if I'm answering this question now, or in 2 years when I do the follow.
How do you see it?
Sorry.
How do you see it? That's my question. I know it can be very.
Yeah. So I see this work moving forward. I want the kind of stuff that I want to do is, for instance, I want to do longitudinal versions of this, and ultimately see if I can use this for mostly prognostic purposes. So one set of results that I didn't show here was that the expression of these measurements, of these multi-dimensional features that I found, some of these measurements could actually predict psychiatric diagnoses that people had at the time of the study. So now this is not, this again, all this data comes sort of retrospectively. It's cross-sectional. So in the future, I'd like to do this longitudinally and see whether we can actually, based on scans at time 1, predict clinical outcomes at time 2. So in that sense, I would say that this is more applicable to prognostic purposes.
Okay.
Now, it's an interesting question that you ask, because really, perhaps you may have picked that up from what I was saying towards the end. I don't fully agree with this idea of diagnosing people as concussed, non-concussed. I think that distinction really doesn't do much, and I think that the future clinical trials will be to stratify people based on these multidimensional features.
Okay, super. Thank you. Great work.
Thank you.
Thank you. Just to follow up with Sammy's question, Guido, you talked about the corpus callosum and the importance of looking at white matter in these patients. Can you comment on what they're doing right now in the clinic? I mean, is all this information being lost, or are they just looking at the whole brain?
At the clinic right now it's not even concussion clinics generally, unless there's some more like perhaps some of the more cutting edge ones, if you will. The way that concussions are managed clinically right now is based on symptoms alone. You can't pick up a CT scan and say, there's your concussion. So I think it's about the percentage, about 90% of concussions are invisible to conventional imaging approaches. So nobody clinically, or most people clinically, are not using imaging. They're just using symptoms. And there are several reasons why that's inadequate.
Okay, so that's really helpful, because that's, what we're able to do at IMICA is really use those diffusion MRI scans, look at the white matter, and also look at the subregions. So all this information is being lost. So that's really helpful. My next question comes from Yusuf Osmanelegu. Yusuf, go ahead.
You hear me well now?
Yes.
Okay. Oh, thank you, Guido. This is a really, really interesting presentation. Actually, I'm also working on moderate to severe TBI, and we're planning to submit a paper coming Monday. So my question in general is going to be about the concussion biomarker part of your presentation. Because as I was developing a connectomic biomarker, or a measure, I would say, for TBI, our clinician collaborator is saying that, what am I going to do with that? Because let's say you are showing a correlation of 0.3. So what am I going to do with that? It's not that good anyways. If I by just looking at the GCS score, I'm making even a better prognostic thing than what you are suggesting. So how would you, first of all, evaluate how good of a biomarker is that, or where would you like to draw that line for whether to call it a biomarker or not, since this being a big issue for me when I'm writing the paper? Because if I call it a biomarker, then clinician says, well, it really is not. So this is the first question. And then my second question is going to be, our study was longitudinal. So what we are observing was the network structure is changing over time. So in your data set, since you're putting all of them in the same bucket, and most probably they're from different times of like the post-injury, so I think it's likely to affect your results as well. What do you think about this part? And thank you.
Okay, so thank you for the questions. Both are fantastic questions. The first one is, again, a very big question, and depending on who you ask, people have different opinions about this. So I'm going to give you my opinion of what a useful biomarker is. I see when I see these neuroimaging studies, and I treat the word biomarker exactly like your clinician does, I kind of get skeptical, because for me, a good biomarker is one that tells you that fits certain criteria. First of all, it allows you to predict the clinical course, right? It allows you to essentially have good prognosis. And it allows you to do diagnosis, but diagnosis for the purpose of just a yes, no is not enough. For me, you need diagnosis that comes with underlying pathological information, right? What will I do? Say that you can with 100% accuracy classify concussed people from healthy controls. We're no better necessarily at finding cures for concussion, right? Because you don't know what's going on at the level of the brain. So really, I think a good biomarker comes with predictive ability of symptoms and clinical course, and information. It has to be rich in neuropathological information, stuff that actually tells you, okay, So if I'm going to design a clinical trial, this is the drug that I'm going to try to use. And unfortunately, neuroimaging is not there yet, but more and more advanced methods are actually starting to shine light on individual specific pathologies. So hopefully that answers your first question. Your second question is time-since injury. So 100% time-since injury plays a huge role. Another thing that I sort of skipped over was the fact that we averaged out time-since injury. So we regressed out time-since injury. So we statistically accounted for time-since injury, but you can rest assured that had we a data set where you could acquire people, scan people, soon after injury and then later on, that would be better. So that would be preferred, and that's really what we're trying to move towards. We're looking for big data sets, but that control for time-since injury in a conscious way, rather than our post hoc statistical way of controlling for it, because absolutely that will play a huge role.
And actually, in our results, what we saw was time-since injury. The networks of patients were declining over time. They were becoming unlike healthy, but they were having cognitive recovery, and there were no relationship between the cognitive recovery and the change in network structure. So it looks like the plasticity is really happening in gray matter, not in white matter. So, and since we are mostly focusing on, especially in DTI sort of studies, we are focusing on how white matter is changing, and we are trying to use it as a biomarker for prediction. Maybe it's not really working out. So this was also another thing, like it's like a red herring. It's showing something else than what you're expecting to see from it. So this was something to focus on, I guess. I definitely agree that time-since injury makes changes in the white matter pathology, but its relationship with cognitive outcome is not that direct.
Yeah, it's hard to tell though. That's I think one of the points of this study was that it's hard to tell, especially when you're just pooling all the data together, because people are so heterogeneous that perhaps at a group level you're not seeing some of those trends that you expect, but really when you start looking individually, that's where you start noticing these big differences. And I think that was perhaps one of the most important points that I was trying to get across with this talk is people should perhaps stop trying to group all concussion symptoms together as if that was essentially, when you group people together, when you pool everybody together, you get rid of all the stuff that is interesting, and perhaps people should embrace heterogeneity a bit more.
Yep, exactly. This was a very brilliant point. Thanks a lot.
Thank you.
Great, thank you, Yusuf. Guido, I just have one last question. You talked about, the heterogeneity of these patients, and then being able to do this across multiple sites. Data heterogeneity or homogenizing the data is often an issue with multi-site trials. Can you comment on what you think of that, since the population is so heterogeneous?
Yeah, that's an excellent question. So at the beginning stages of this study, we struggled a lot with that because there are certainly, I can confirm that there are site effects that are present in the ABCD study. So there are different ways to account for side effects. The way that we went with was a statistical approach. We regressed out site, but there are certainly much more sophisticated ways of doing that. There are the algorithms with complicated names like Combat and I forgot the name of the other one, but that try to estimate the effects of scanners and remove them from the raw data. And absolutely, the future has to include a way of accounting for side effects, because they play a huge role for sure.
Okay, great. And I have another question from Maxime Decoteau. Max, go ahead.
Thanks, Guido. Amazing talk, great slides. I'm really proud and happy to see this. Maybe to pick your brain a little bit more on stratifying and clinical trials and future of clinical trials. Mm-hmm. These trials are already very expensive, but is in your mind, when you talk about better stratification, do we need an MRI exam before we even get the clinical trial going to group our studies like, how do you see this in the future, and what is it? What will have to be practical, right? This needs to be practical, but the costs are so high anyway that maybe an extra MRI just to recruit and group people properly makes sense. But I'm curious to hear your thoughts, and also your thoughts as a future clinician. Go ahead.
Okay, all right. So that's a great question, and one that I've struggled with since pretty much the beginning of this. This idea that you can develop the nicest and most sophisticated MRI biomarker that exists, but you can't scan everybody. We often in the hospital we see patients from remote communities, and we can't firmly say we're going to scan absolutely every single person with a concussion. I think that the way forward ideally would be to find a practical solution that doesn't require an MRI, but that is still MRI informed. So what I mean by that is that when we're doing this approach in which we're finding these underlying patterns of connectivity and symptoms, what we're creating are these there's two ways of calling these. We're creating these, if we look at symptoms alone, we're creating these patterns of symptoms that are related to some underlying neuropathology that is informed to you by the MRI. So I foresee in the distant future, if we are able to validate all of these approaches and really make sure that they're consistent across different groups, and at some point the MRIs might not be all that necessary, because once we've designed these multivariate features, we can feed in new data into this. And that's part of what we did in this paper at some point. So we create these multivariate features with a large proportion of the data set, and then with a smaller proportion of the data set that we had separated since the beginning and that didn't go through the analysis, we fed that into this algorithm. And that's an interesting way forward, because you could foreseeably do this without even actually having to acquire an MRI. Just feed the symptoms into these multivariate pairs, and it'll tell you how much you express the multi-symptom part of that pair, and that should, in theory, also tell you something about what's actually going on at the level of the white matter connectivity. So ultimately, absolutely, after everything is perfectly validated, the way forward can't be to scan everybody. We still need a good way of using the clinical data we do have to design these clinical trials.
Yeah, and leverage the existing MRI data, as you mentioned.
Exactly.
Maybe just a last, I'm curious to hear you about maybe another hot topic in North America is sports-related concussions.
Mm-hmm.
And having a quantitative score that could help physicians and clinicians and sports therapists help teams to know when a player can return to play or not. What's your vision on this, and do you see that such a multi-track, multi-symptom analysis could help there as well?
Okay, that's an excellent question, because that ultimately it touches almost upon philosophy, and what I mean is that there's more and more studies now that what they're doing is that they're scanning people soon after the injury. These are sport-related concussions, right? Soon after the injury, and then at the time that their physician clears them to go back to whatever activity, whatever sport they were doing. And what these studies are finding time and again is that at the level of the brain, there are still abnormalities, right? And some people fear that that's what's driving these horrible cases of chronic traumatic encephalopathies, the fact that people are routinely cleared to return to the activities that caused their injury while their brain is not healed. The reason why I say that this touches almost upon philosophy is that there's a serious discussion among clinicians as to what do these brain abnormalities mean? What does a brain abnormality that is not accompanied by symptoms, what does that mean? Is that really subtle brain abnormality? Is that subclinical? So I think there's so much more discussion to be had about that, about the meaning of these subclinical brain abnormalities. And one thing that I'd be interested, because you asked what's the role of these multi-track, multi-symptom features. definitely in the future, I'd be interested in doing a study in which we perform something similar. We look at people once they're cleared to return to play, and we look at whether on these multi-dimensional connectivity features, whether perhaps that's where they show actual healing that goes with symptoms, or rather perhaps the reverse. Perhaps we scan these people when they're cleared to return to play, and we find that they have abnormalities that point towards very subtle symptoms that the clinicians perhaps were just not paying attention to. But there's just so much to do with this data, that's for sure. With this approach, rather, that's for sure.
Thanks, Guido.
My pleasure.
Okay, great. Well, thank you very much, Guido. Really appreciated your talk, and thank you to everyone for attending. So if you could please fill out that brief questionnaire at the end, that would be really helpful for future webinars. So again, I'd like to say a final thank you to everyone, and I hope you all have a great day. Look forward to seeing you on the next webinar. Bye now. Bye, Guido.
Thank you. Bye-bye.